Hello, and welcome to everyone joining us for today's Select Science webinar titled Enhance Productivity and Purity in Next Generation Bispecific Antibody Manufacturing. My name is Matilde Marques. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Dr. Heng Tai Liew, Senior Protein Scientist at GenScript Biotech. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of Auditorium and click to open the Q&A window. So, without further delay, I'm delighted to hand over to Dr. Heng Tai. I'd like to thank him for presenting to us today.
Good morning, everyone. I'm Heng Tai, a senior protein scientist from GenScript. Thank you all for joining us today. I'm truly excited to welcome you to this webinar focused on one of the most promising advances in the biopharmaceutical manufacturing, the next generation bispecific antibody production, and more specifically, how TurboCHO technology is helping redefine the efficiency and scalability in this space. As we know, the development and commercialization of bispecific antibodies have opened up remarkable therapeutic possibilities, especially in areas like oncology and autoimmune diseases. However, with these opportunities come new challenges in manufacturing, from expression efficiency to the purification complexity. That's where TurboCHO comes in. Today, we'll explore how this next generation platform not only boosts the productivity, but also helps streamline the downstream processing, enabling faster, more cost-effective delivery of high-quality therapeutics. Whether you are involved in process development, manufacturing, or analytical sciences, so this session aims to offer valuable insight you can apply to your own work. So we'll also be taking your questions at the end of the presentation, so please feel free to drop them into the chat as we go along. Let's dive into the future of bispecific antibody manufacturing, efficient, scalable, and ready for tomorrow's therapeutic demands. Bispecific antibodies are a diverse class of the molecules engineered to recognize two different epitopes or antigens simultaneously. With over 700 bispecific therapeutic pipelines currently in development and 75% still in the R&D or the preclinical stages, the field is rapidly growing, but also faces significant manufacturing and development challenges. By simultaneously targeting two distinct antigens or epitopes, bispecific antibodies can trigger multiple physiological or anti-tumor responses, either independently or in a coordinated manner. Functionally, they can mimic the effects of combination therapy using two monoclonal antibodies, but with the advantage of being a single engineered molecule. This not only simplifies manufacturing but also reduces treatment complexity for patients. Additionally, their synergistic effect may also enhance the therapeutic efficacy beyond what individual antibodies could achieve alone. Currently, more than 700 bispecific antibodies are in clinical development, with the majority still in the early phase trials. So the FDA has approved more than 10 bispecific therapies for the treatment of cancer, as well as hepatology and ocular conditions. Bispecific antibodies format can be categorized into three types: the asymmetric, symmetric, and the antibody fragments. The choice of the types depends on your therapeutic goals, target antigens, and desired pharmacokinetics and the pharmacodynamic properties. Each bispecific antibody type offers unique advantage and considerations for engineering, manufacturing, and clinical use. You can scan the QR code here to access the interactive production map to see our production insight for 70 plus different formats. So in the map, we have rated each format by its manufacturability score and the recommended purification steps. The production of bispecific antibodies presents several complex challenges across multiple stages. First, the design and expression stage include the challenges such as the secretion issues and the N-terminal miscleavage of sigma peptide, which can affect product quality. Additionally, achieving balanced expression of each antibody chain is also critical, as imbalances can reduce overall yield and functionality. Moreover, optimization of the expression hosts and also the vectors is essential to enhance the titers and ensure the efficient, high-quality production. In the next stage, purification poses difficulties due to the presence of the multiple impurities and the need for the selective removal of the mispair or incomplete species. Finally, the quality control and the characterization are critical to verify the structural integrity, binding specificity, and overall the product consistency. So the unique challenges due to their complex structure, which complicate the analytical processes. Additionally, the slight molecular weight difference between the mispaired species and the product of interest make it difficult to effectively separate the impurities, requiring advanced and high selective characterization techniques. So, to successfully optimize bispecific antibody production, a comprehensive and strategic approach is essential. First, molecular design plays a critical role in minimizing the impurities such as the chain mispairing and aggregation, setting a strong foundation for the product quality. Next, the expression optimization involves fine-tuning multiple factors, such as the cell lines, the vectors, the culture media, and the cultivation processes, which carefully balancing the chain ratio according to the unique characteristic of the molecule. Moving on, the purification optimization leverages the subtle difference between the product of interest and major byproducts, guided by an intelligent purification method recommendation system to achieve efficient and selective separation. Finally, ensuring the optimal purity and yield depends on robust analytical methods and advanced mass analysis technique to accurately identify the target product and any impurities throughout the process. Together, so all these strategies enable us to overcome the inherent complexity in the bispecific antibody manufacturing, delivering high-quality therapeutics more efficiently and reliably. So how can we reduce the mispairing and impurities right from the design stage? Common impurities include the homodimers, half antibodies, like chain mispairing, three-quarter antibodies, and species formed through the non-covalent interactions by carefully engineering the molecular design, such as the optimizing chain mispairing interfaces and incorporating stabilizing mutations. So we can significantly minimize these unwanted byproducts. So this proactive approach helps improve the product quality and simplifies downstream processing. So the molecular design included such as the knob into hole, cross-mat, electrostatic stirring, and also the in vitro assembly. One effective strategy to prevent the mispairing is the use of the dual map approach, which is going to be shown over here. So dual map improves the efficiency of the correct heavy and the light chain pairing by replacing the native disulfide bond in one of the CH1 and Cl interfaces with an engineered disulfide bond. So this modification stabilizes the pairing between the connect chains, significantly reducing the mispairing and enhancing the overall quality of the bispecific antibody. At GenScript, we employ a cutting-edge integrated approach to optimize the bispecific antibody production during the expression stage. We begin by using a single-cell printer combined with the cell metric imaging technology to precisely isolate and monitor the individual cells, ensuring the selection of the healthiest and the most productive clones. So this process is supported by an automated workstation and high-throughput detection system, so which accelerate the screening and enable rapid identification of the top-performing clones. Once the best clones are identified, we apply advanced cell engineering techniques to enhance the expression characteristic, followed by the development of the optimized cell pools. So this streamlined workflow ensures the high productivity, consistency, and scalability, laying a strong foundation for efficient bispecific antibody manufacturing. Another key part of our optimization strategy is the model and data-driven process optimization. So this involves using the design of the experiment, the DOE, to systematically explore critical variables and their interactions. Coupled with the model-guided analysis, we can also interpret the complex data to identify optimal conditions for expression, purification, and overall processes performance. So this approach accelerates the development, reduces the try and error, and leads to more robust, scalable manufacturing processes. Since the launch of TurboCHO 1.0 in 2021, we have seen a significant increase in achievable yields with the upgrade to TurboCHO 2.0. Even more exciting, TurboCHO 3.0 is set to launch by the end of this year, promising a major leap forward with even higher titles and enhanced production capabilities. So this continuous innovation underscores our commitment to pushing the boundaries of the bispecific antibody manufacturing. As shown in the figure below, the yield improvement with TurboCHO 2.0 upgrade significant compared to the 2.0 version, highlighting the substantial advancement we've made in enhancing the productivity. Beyond optimizing cells and vectors, chain ratio optimization plays a crucial role in addressing the imbalanced expression of each antibody chain in bispecific antibody design. Since each chain naturally expresses at different levels, adjusting their ratio can significantly reduce the impurities caused by this imbalance, ultimately improving product quality and yield. As one of the case study, we found that by overexpressing the chain which do not bind to the resin, allow for high purity obtained in a single purification step. From the SDS page, the higher the ratio of the FabFC, the higher the purity. So please note that the FabFC chain here contains the double mutation H435R and the Y436F, which can prevent the binding of the FC to the protein and resin. For the slide here, it shows that the chain ratio not only affects the purity, but can also be used to improve the expression yield of the target antibody. Having a roadmap for the bispecific antibody purification is essential because of the complexity and variability involved in producing these molecules. Unlike standard monoclonal antibodies, bispecific often have multiple chain formats and can generate a range of product-related impurities, including mispaired species, fragments, and aggregates. A well-defined purification roadmap helps guide the selection and sequencing of the purification steps, such as the affinity capture, ion exchange, based on the specific characteristic of the molecule. This structured approach enables developers to maximize purities and recovery, streamline processes developments, and reduce time spent on try and error. So it also ensures scalability and consistency as the molecule moves from the development to the clinical and the commercial production. In short, a purification roadmap transforms complexity into a controlled, predictable processes. For example, the common polishing step for bispecific antibody depends on the format, with the SEC or the IEX for the aggregate remover for non-IgG-like bispecific antibody and symmetric IgG-like bispecific antibodies. Other methods such as the AC, IEX, HIC, or MMC will be served as the potential polishing step for the asymmetric IgG-like bispecific antibodies. To streamline purification, we leverage a chromatography strategic recommendation system in GenScript, a data-driven approach to guide method selection. So this system enhances decision consistency and promotes the operational standardization, ensuring that purification strategies are based on the objective criteria rather than try and error basis. By minimizing human intervention in both the selection and the execution phases, we reduce the variability and increase the efficiency. Importantly, This system supports continuous iterative optimization, allowing us to refine the purification conditions over time and adapt to the specific needs of each bispecific antibody molecule. For example, the first step purification like affinity chromatography is determined by the sequence, and the second steps and onwards are determined by other factors such as the isoelectric point, the molecular weight, hydrophobicity, and binding strength. Here are a few cases I will be sharing in the next few slides regarding how GenScript optimizes the purity from different aspects due to the characteristic of the bispecific antibody, and most importantly, the consistency throughout the production line, such as the specificity to resin, hydrophobicity, isoelectric point, etc. The first case study here refers to show the design drive the choice of the purification method. The heterodimeric IgG contains two different light chains, like lambda and the kappa light chain. Some common strategy, like the protein A, may be implemented by the human decision. In this case, will result in the mixture of the light chain mispairing in the products. But GenScript Streamline platform suggests the map select VL to remove the light chain mispairing construct, which result in a good separation of the light chain mispairing products from the target antibody. Another case involved the heterodimeric IgG. After the first step protein A purification, impurities such as the aggregates, three-quarters antibodies, antibody without two light chains, and half antibody were detected. So the streamlined purification platform in GenScript suggested Heng as the polishing step according to the impurities, resulting in a nice separation of target antibodies from the impurities. The third case involved the separation, which is based on the difference of the binding strength of the target antibody and impurities to the resin. After the first step protein A, antibody losing one and the two light chains was detected, resulting in low purity. According to the variation in binding strength of the impurities to the map select VL resin, we have managed to separate the target antibody from the impurities losing the light chains. Another step of the size exclusion chromatography further removed the covalent aggregates, therefore resulting in high purity of the target bispecific antibody. Last but not least, the final case here is focused on the isoelectric point of target antibody and impurities. How the platforms analyze different impurities, isoelectric point, and design a purification strategy to tackle the challenges in this purification protocol. After the first step protein A purification, aggregates, three-quarters, and half antibody were detected. Based on the Heng reverse phase MS analysis, we identified the light chain mispairing, which is not observed from the SDS page analysis. Cation exchange coupled with the pH gradient was then designed according to the isoelectric point differences, therefore resulting in remarkable separation of the target antibody and the impurities. As we have explored, the path to success bispecific antibody manufacturing is complex, but with the right strategies, is absolutely achievable. From the molecule design stage to the expression optimization using tools such as the single-cell printing, high-throughput screening, and the cell engineering, every step matters in driving the efficiency and product quality. We have also highlighted the importance of the chain ratio optimization to balance expression levels and reduce impurities, and how model and data-driven approaches, including DOE, to help accelerate the process's development. On the purification side, having a clear roadmap is essential to navigate the structural complexity, and our chromatography strategy recommendation system ensure the consistency, reduce the human bias, and allows continuous refinement. With the evolution from TurboCHO 1.0 to 2.0 and the exciting upcoming launch of the TurboCHO 3.0, we are seeing remarkable gains in the yield and the scalability. So this progress is not accidental. It's the result of the continuous investment in the R&D and production process optimization, all aimed at creating a stronger, more reliable platform for the bispecific antibody development and manufacturing. As we look ahead, we remain committed to pushing the boundaries of the innovation, so working alongside our partners and the broader scientific community to make the high-quality biologics more accessible, efficient, and impactful. Thank you again for being with us, and we look forward to continue this journey together. That's all for my presentation today. Thank you for having us today.
Thank you, Dr. Heng Tai, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We will now move on to the last part of today's webinar, the Q&A session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of auditorium and click to open the Q&A window. So, to kick off this final portion of today's event, the first question I have for Dr. Ang is, how does the platform ensure correct chain pairing in complex bispecific antibody formats with asymmetric light or heavy chains? And how is this confirmed analytically?
Yeah, thank you for the questions. Actually, like GenScript ensure the correct chain pairing. So especially like in the complex bispecific antibody formats, we actually employ a variety of the advanced engineering strategies. So for example, like the KIH for promoting the heavy chain heterodimerization or cross map. To actually enforce the specific light chain pairing. So of course, like the use of the common light chains in formats such as like the dual body to simplify the assembly. Additionally, just now that we actually also mentioned the dual map. So you actually leverage the CH1 CL match interface to guide the correct pairing without the need like for the extensive mutation. So these design approaches are actually supported by the analytical validation, like including like the mass spectrometry, the SEC, HPLC, the SDS page, and of course like the peptide mapping. So to ensure the correct assembly. So yeah.
Lovely, thank you.
Mhm.
And how universal is the TurboCHO system across diverse bispecific formats? Can it handle FC-based and non-FC-based designs, tandem single-chain variable fragment formats, or VHH fusions with similar performance?
Yeah, actually, the TurboCHO is actually a very versatile like expression platform. So you actually like that's demonstrate like a very broad applicability. So across like a very diverse like formats for the bispecific antibody. So it also like support not only the FC base but also like more importantly, so which is the non-FC based design. So like including the tandem SCFV formats, like also the BITE and also like the VHH, the nanobody fusions, and of course like with a high expression use. And the most importantly is the consistent quality.
Perfect, thanks. Next question says, what yield targets in grams per liter are typically achieved for complex bispecifics with multiple domains or flexible linkers?
Yeah, especially, so I think this is a very good question. So, like for complex bispecific antibody, so especially those like with multiple domains and flexible linkers, a very long linker design. So, GenScript expression of the TurboCHO platform actually can typically achieve like the use in the range about like 1 gram per liter or above, like in the transient expression system settings like for let's say for the optimized constructs or in let's say in the stable cell line development, so the use can even be higher. So actually it all depends on the format and as well as the expression strategy that we have. So like for example, we know that like some complex bispecific formats, right? So especially like the tandem SFV or VHH fusions, those with a very flexible linkers, so they actually tend to have a lower expression efficiency. So because of the molecular complexity. But however, with the GenScript like corner optimization, so of course, like just now we mentioned the vector engineering, so all these can actually help to mitigate like these kind of limitations. So yeah.
Lovely, thank you.
Mhm.
And does the platform use single chain Fab or dual variable domain approaches to eliminate the risk of incorrect pairing altogether?
Mm, yes, the GenScript platform actually support like the single chain fab, so and also like the DBDIG, so we call the dual variable domain. So yeah, all these actually can be used to eliminate the incorrect like chain pairing. So in the bispecific antibody production, so as we know that for let's say for the single chain formats, right? So it actually links the variable and the constant region, so of the heavy and light chain into a single polypeptide by using the flexible linkers in case like some audience actually don't know what single chain fab. So all this is actually can use to ensure the correct intra molecular pairing. And also, the DVD-IG approach that we actually can also do is actually they just stacks two variable domain. So on each arm of the IgG, so then they can actually allow dual antigen targeting within a single unified heavy light chain structure. So as we just now we mentioned, both formats can actually reduce the mispairing risk and are actually we constantly actually incorporate into the design that actually can help to prevent like the mispairing.
Thank you. Um, Next question says, does the purification platform enable effective separation of correctly paired bispecifics from mispaired species? And what chromatography techniques are most effective? For example, mixed mode or caption exchange?
Yeah, so we actually the GenScript purification platform. So it's actually designed to like effectively separate the correctly paired bispecific antibody, so from the mispair or the unwanted species. So using actually we use a series, a combinations of the chromatography techniques. So like the purification process, like for the bispecific, typically like in start from let's say the protein A affinity chromatography. So for the initial capture of the Fc containing bispecific antibody, then after that they follow by the polishing steps, such as for example the just now you mentioned the cation exchange chromatography and also the mixed mode chromatography. So can be also used to like resolve like those very small differences in charge or like the hydrophobicity and also like the conformation between the correct target bispecific antibody from the impurities. Yeah, so like probably in this case study that we actually just now we show, right? So the CEX is actually particularly useful in separating the charge variants like from the mispaired species due to their actually surface charge profiles. Yeah.
Thank you, Dr.
Hmm.
Eng. Is the purification process already standardized, or is it customized for each antibody format? And how might that change in the future?
Yeah, so this is actually a very good question. So let's say the GenScript purification process, like for the bispecific antibodies. So I would say like it's actually more like a hybrid approach. So we actually combine the standardized workflows and with the format specific customization. So that means that we have the standardized workflows, but simultaneously we also have the format specific customization. So let's say just now we mentioned, so the platform we actually employs a core purification step. Just now we mentioned the protein A affinity chromatography for the Fc containing molecules, and then like also the commonly used polishing step like the CEX or the mixed mode chromatography. So these are actually broadly applicable to across many bispecific antibody formats. But however, so due to like the structural or the biophysical diversity of the bispecific antibody, so like for example the SAFE base, etc. So the polishing strategy have to be like custom tailored so to those specific molecule, so to actually optimize the purity and the yield. Yeah, so I think in the future, so like the bispecific antibody, the purification technologies will actually become more and more mature, and more data are actually can be collected across like the different formats. not just GenScript, the industry is actually expected to move toward I would say more semi-automated purification platform. So and then the those validated like purification template can be actually rapidly adapted to like the new bispecific antibody construct. So with like minimal reoptimization. Yeah.
Lovely. And are new affinity ligands, for example, FC variant binder, under development to enhance purification selectivity for bispecific antibodies?
Yes, for the new affinity ligands. So actually, GenScript and like broader bioprocessing field, they're actually actively developing like the novel affinity ligands, so to actually enhance the purification or correctly pair bispecific antibody. So, for example, let's say the CH1 domain specific resins, right? So, like the Presto 70 CH1 and the Presto 850 high pH resin. So all this actually allows the illusion of the bispecific antibody at a milder pH, because all this can be used to avoid like the pH sensitive aggregation. But GenScript actually also offers a specialized like affinity resin. So for example, like the anti-jes linker affinity resin. So all these can also specifically binding to the proteins like contain the jes linker. So and then which are also like commonly used in the bispecific antibody, and especially like those SCFV, so tandem VHH and other fusion proteins.
Thank you. Um, what specific challenges exist in removing tightly bound host cell proteins, DNA, or structural similar impurities in current process?
Yeah, so the HCP and the DNA, so all these are actually quite common. So, like the challenge in the bispecific antibody production. So, which actually we not actually didn't mention much about this, like in the slides just now. So, like for example, the HCP. So, because I think the biggest challenge is that they will actually form the stable non-covalent interactions with the target bispecific antibody. So, especially during the protein expression. So and then another nightmare is that this can also like co-elude with your protein A capture, and then they can be also like found in the remain through the polishing steps. So what we can actually do for this case, for example, so we can actually perform like the anion exchange, like for example, also like some multi-model chromatography. So all these actually can use to actually wash off the HCP from the target bispecific antibody. So all these actually can be there are actually like some protocols which can be also like found in the previous literature, and then also like for the host cell DNA, so it's another challenge because like sometimes the DNA can like bind electrostatically like into the positive charge region of the bispecific antibody. So especially like in the very early step. So like we actually also have like come develop some protocols like the ion exchange, the anion exchange chromatography. So all these actually can be used to like remove like all these like impurities. Yeah.
Great. And why might some impurities be invisible or hard to detect with standard gel tests?
Yeah. So regarding these questions, so like why there are some impurities, they are actually can you actually repeat the questions again?
Of course, of course.
Mhm.
Why might some impurities be invisible or hard to detect with standard gel tests?
Yeah, so why they are actually can't be detected, right? So, for example, so if they can't be detected, so that means that actually have a very similar molecular weight, like with the target bispecific antibody. So in this case, so they will actually co-migrate with your target bispecific antibody, and this could be actually very challenging. So as that means that uh, you actually can't use like the purification strategy like the SAC to actually further separate it from the target bispecific antibody. And then in this case, and you also need like to have some more advanced like analytical techniques, like for example the mass, or just now we mentioned the two-dimensional mass. All these can be used to actually detect the presence of these like mispairings or the impurities. Yeah.
Thank you. And how do labs make sure they are not missing important impurities when testing complex molecules like bispecific antibodies?
Yep. So for example, like in this case, what we can actually not make sure, or to make sure that there are no mixing like important impurities, was when we actually test the bispecific antibodies. There are actually several things that we actually can do. Just now we mentioned the mass spec. So and then also like what we can do is the probably the SDS page, the CSDS, and also like sometimes the seg mounts. Yeah, they can actually allow us to detect the size variance and also like the aggregates. So all these analytical methods, so they actually can be used like I mean like in combination. So to actually make sure that you don't actually miss the important impurities.
Well, thank you, Dr. Hang. I think we still have time for one more. And why do trispecific and multispecific antibodies present more analytical challenge than bispecific antibodies?
Yeah, so this is actually a very interesting question. So in this case, like the trispecific or the multi-specific antibodies, right? So they actually often require, let's say, I would say, two, three or more like antigen binding domains. So in this case, that means that they will have multiple chains. So probably like four chains, five chains, or even more. So all these will actually lead to a very large number of the heavy chain or the light chain combination. So that means more chance. This will actually increase the risk of the incorrect chain pairing. So and the also like all these they actually can make the target bispecific antibody and the impurities harder to be detected and resolved. So in this case, yeah, so that just now I mentioned the chain. So the number of chains is actually definitely one of the biggest challenges if we actually proceed to like the multi-specific antibody. So also like as we know, so if you actually have more chance, like and even like some chance that actually probably have like multiple GS linker. So if you actually have a very flexible GS linker, all this can also like cause the truncation. So the truncation may actually happen, and then if the truncation happen, this can make your bispecific antibody production even more, multi-specific antibody production more challenging. So and then the more difficult part is actually the analytical part. So because if you actually have more chance, right, so then how do we actually make sure that like the method or the mass spec that actually you have is actually qualified or sufficient to actually resolve like the let's say what's really inside in the products or the mixture of the impurities and the products. So all these actually you require like a higher dimension of let's say the mass spec actually to resolve this. So and also like the charge. So all these things they can also like complicate like the I mean the productions of the multi-specific antibody. Yeah.
Thank you so much, Dr. Hang. I'm afraid that is all we have time for today. I would like to thank Dr. Heng Tai for today's discussion and presentation and for this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you found this a worthwhile session. If we didn't quite get to your question today, we'll follow up with answers to these after the webinar. And if you have any other questions after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And for those watching on demand, a certificate can be requested by emailing us at the same email address. And if you like to listen again to today's webinar or invite a friend to do so, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.